My name is Janice Young. I am a senior staff scientist at Life Technologies, where I've been since 2006. I work in the medical sciences R&D group, and I will discuss companion diagnostics opportunities with you today using our technology and strategy. I would like to mention at this time, when questions come to mind during my talk, you should submit those questions by typing them into the Q&A window on your screen. The title of today's talk is Companion Diagnostics Co-Development. Are you ready? We will look at some of the factors involved in co-development of a companion diagnostics test. Many decisions are made long before development of a diagnostic test begins. A biomarker test using off-the-shelf assays in the research lab may have potential as a diagnostic. And then what? What are the key considerations in choosing a companion diagnostics partner? First is technology. Second, experience in diagnostic development, such as design control, regulatory expertise, manufacturing. Third is global commercialization strategy. And fourth is competent management of the companion diagnostics partnership. Go to slide 2. I'm going to tell you a little bit about our company, and I will talk for about half of the talk on technology, and then the second part will consist of companion diagnostics topics. Our company is headquartered in Carlsbad, California. We were incorporated as Life Technologies, formed by the merger of Applied Biosystems and Invitrogen in 2008. Currently, we are traded on the NASDAQ stock exchange under the Life ticker symbol. And as you have probably heard, we plan to merge with Thermo Fisher, subject to regulatory approval next year in 2014. This slide shows our businesses and the diversity of life sciences served by our company. First, we are well known for providing tools and instruments for solutions in basic and translational research. Second, with these capabilities, we are committed to developing diagnostic applications in molecular medicine, and this will be the bulk of what I am discussing today. Third, our forensic testing kits are used to solve crimes and also in human identification. For example, this technology was used to reunite families who were separated after crimes of human trafficking. The fourth business is our food and water safety. This was used for monitoring at the last Summer Olympics by the UK's Health Protection Agency to ensure food safety. And then lastly, synthetic biology. What is that? It combines molecular biology and systems biology with applications in biofuels, industrial enzymes, biocloning, and gene synthesis. Our mission statement is to make life even better. What Technologies brands are shown here? It's a little bit like introducing the starting lineup of a baseball team. So I'll start with the leadoff brand. When we merged with Invitrogen, the Applied Biosystems brand was still retained, and that brand name remains on our products today. So Applied Biosystems instruments that were manufactured under the Applied Biosystems Incorporated company, for example, still will bear the AB brand, but they are supported under Life Technologies. Second up is the Ion Torrent brand, rather small to see. Ion Torrent uses semiconductor technology to deliver the fastest benchtop next-gen sequencing. Third up Next to Ion Torrent is the Novex brand, which has reagents and consumables for protein separation. Fourth up is the TaqMan real-time PCR brand, which uses probe-based chemistry for quantitative gene expression, genotyping, copy number, and mutation detection. Fifth up is Ambion. Ambion provides high-quality, innovative RNA products for purification and transcriptome analysis. Next up is Molecular Probes, which has novel fluorescent reagents for flow cytometry and other cell biology applications. Seventh is Gibco. Gibco supplies cell culture media for the highest quality, consistency, and performance for reproducibility in cell biology and cell-based technologies. And then finally, branding eighth is Invitrogen, who was a pioneer in molecular biology, providing cloning and expression kits, and who is still well known in providing tools for genomics and proteomics research. So you can conclude from this that LifeTech's business is product development. Our product commercialization process is used for reaching checkpoints in development, manufacturing, and product launch. This capability, as you will see, extends to companion diagnostics development. Our next corporate slide shows quick facts about Life. We are a global company with presence in over 180 countries. We have over 1,500 scientists, over 10,000 employees, with 600,000 plus citations in the literature using our products, and over 4,000 patents issued and pending. Our customer base is over 75,000, with $3.7 billion in sales for 2012. This slide shows the markets served by our business. Currently, LifeTech is organized in 3 divisions: research science, applied sciences, and medical sciences. As I've described, the research science division has tools for molecular and cellular biology, and also is an umbrella for the genetic analysis platforms such as quantitative PCR and TaqMan, CE sequencing, and next-gen sequencing. Applied sciences are represented by synthetic biology, food safety, animal health, human identification, bioproduction, and agriculture biotechnology. Medical sciences is the umbrella for molecular diagnostics as well as genomic medicine and regenerative medicine such as stem cell biology. 3 recent acquisitions in medical sciences show LifeTech's commitment to molecular diagnostics and medical devices. All in 2012, I'm going to describe 3 acquisitions. Last October, LifeTech acquired Companion Bioscience, a preeminent cancer bioinformatics company founded by Arul Chinayan and Dan Rhodes. The services of this company are widely used by pharma to identify novel gene targets for drug discovery and development. Companion has sequence data from over 62,000 cancer patients. Its deep oncology expertise and high-value proprietary assets enhance Life Technologies' diagnostic development capabilities across multiple platforms, including next-gen sequencing, qPCR, and proteome analysis. The second acquisition I will describe is Pinpoint Genomics and their CLIA-approved lung cancer test. To read more about this, go to a Lancet publication which appeared last year entitled "A Practical Molecular Assay to Predict Survival in Resected Non-Squamous Non-Small Cell Lung Cancer, Development and International Validation Studies." The third acquisition was Navigenics, which expanded Life Technologies' capabilities in diagnostics. This next slide has examples of our technology as applied to diagnostics tools used to provide solutions. Our diversified product lines are instruments and reagents, which we provide, as you can see on the left, as OEM. That stands for Original Equipment Manufacturer, where we supply these tools for research use only. In addition, many of these are sold to other companies who retail those products under a third-party purchasing company's brand name. So for example, Dynol beads, those are used in more than 25,000 IVD instruments. So these beads are optimized for immunoassay IVD, molecular diagnostics, and nucleic acid IVD. They have features well suited for diagnostic assays such as fast kinetics and high capacity, high signal-to-noise ratio, quality, and lot-to-lot reproducibility. Molecular Probes is a well-known brand for cell labeling and detection used for fluorescence imaging, flow cytometry, and cell viability assays used in clinical labs. Finally, Tropics substrates are used as chemiluminescent substrates for detection of alkaline phosphatase in ELISA and reporter gene assays for general lab use as well as IVD. In the middle, under Lab Tools, are examples of Life Technologies instruments. The AB3130 is used for fragment analysis and sequencing in clinical labs for diagnostic virology testing and microsatellite-based genotyping. And it can also be used as an open platform, allowing for custom test development. Right in the middle there is the Verity DX thermal cycler, which is the first Applied Biosystems endpoint thermal cycler for in vitro diagnostic use. The Verity is classified as a U.S. FDA Class 1 medical device. It conforms to IVDD requirements and is CE IVD labeled in Europe. The instrument is manufactured to ISO 13485 and GMP requirements. The instrument is available in 3 formats for thermal cycling: 96 well, both standard and fast, and standard 384 well. All 3 systems feature an exceptionally easy-to-operate color touchscreen to simplify instrument setup and use. This instrument is available in the U.S., Europe, and other selected countries. The last tool that's shown in the middle is Acrometrics. Acrometrics is a company which LifeTech acquired in the year 2010, and it offers a range of high-quality molecular products designed to meet customer needs. The product range includes controls and standards to assist in the implementation and monitoring of diagnostic assays for clinical labs, blood centers, and manufacturers. On the right are our regulated solutions for IVD products. First, the 7500 Fast DX instrument. It has a regulatory 510K clearance as a clinical multiplex instrument. Second, Life Technologies launched the CE IVD marked QuantStudio DX for quantitative PCR last November for clinical diagnostics labs in Europe. In addition, in March, the FDA cleared the Quidel C. diff assay for our QuantStudio DX and the 7500 DX. The instrument can perform diagnostic functions, including pathogen detection, gene expression analysis, SNP genotyping, copy number analysis, mutation detection, microRNA and other non-coding RNA analysis, and high-resolution MELT analysis. Third and finally, in February of this year, the FDA cleared LifeTech's 3500DX genetic analyzers and the C-Core HLA typing kits, the first 510 cleared sequence-based system for HLA typing in the U.S. Also, the 3500DX is now the only 510 cleared Sanger sequencer commercially available for the diagnostics market. LifeTech's worldwide install base of real-time PCR instruments is greater than 28,000 units. Of the 28,000 instruments in labs around the world, approximately 7,500 are placed in labs, in clinical testing labs. So if you look at the horizontal running through the middle of the slide, this shows the continuum of discovery to characterization to screening. LifeTech instruments and tools cover this spectrum. The vertical arrow from top to bottom on the right shows the continuum of RUO to IVD. Thus, the instruments above the horizontal line or arrow are for RUO. But in addition, we offer diagnostic platform solutions customized for the number and type of analytes with simple and reliable workflows amenable to IVD. So those are the instruments that are below the horizontal arrow. I hope you will see that this demonstrates LifeTech's commitment to both research and diagnostic testing. If you look at the left, there is a photo of our next-gen sequencing instrument, the Ion Torrent Proton, which is for genome scale discovery, largely for research. Next to it is the Ion Torrent PGM, or Personal Genome Machine. And this is, as you can read, for targeted sequencing, resequencing, and expression analysis. Note that the Ion Torrent is not currently cleared by the FDA, but it will be ready for diagnostic purposes in the near future. If you would like to know more about Ion Torrent technology and applications in biomarkers and diagnostics, you can listen to the presentation given yesterday by Brian Kelly from LifeTech entitled Unlocking the Potential of Precision Medicine with Ion Torrent Semiconductor Sequencing. Next, capillary electrophoresis in the middle of the slide is useful for discovery, and also it provides unequivocal evidence of somatic mutations, specifically when the mutations or alleles in a region of interest are not known. This technology can be advantageous. However, it does not detect deletions of an entire exon or amplifications, and in the case of mutation detection, CE sequencing requires the mutation to be present in at least 20% of the DNA samples, in at least 20% of the DNA templates in a sample. It is useful for characterization and screening of polymorphic loci. And as I mentioned, in February, LifeTech received an FDA clearance for the 3500DX genetic analyzers and for its C-Core HLA typing kits. LifeTech received the 510K clearance for the 3500DX and the QuantStudio DX PCR system this year. These two systems have been designed specifically for use in the clinical testing laboratory and will serve as LifeTech's flagship diagnostic instruments for qPCR and Sanger sequencing, respectively. All the way to the right is represented our TaqMan assay platforms, which include over 1.2 million gene expression assays for human and model organisms, 1.6 million human copy number assays, 4.5 million human SNP assays, and 2,700 human DME genotyping assays. Also included are 12,000 non-coding RNA, and finally, over 700 mutation detection assays. Our platform of mutation-specific assays are called TaqMan qPCR. So qPCR stands for Competitive Allele-Specific TaqMan PCR. This is a cost-effective solution, and in most cases, it is more sensitive than sequencing when tumor cells represent a minor fraction of the cells in the clinical sample. So for example, the qPCR assays can detect 10 copies of mutant in the background of 10,000 wild-type copies, which is a sensitivity of 0.1%. The set of approximately 800 TaqMan qPCR assays are designed for 46 genes with hotspot mutations. These are now commercially available, and they have been actually since last year. So our off-the-shelf assays have been tested and optimized. If we are missing a marker of interest, we are able to run the sequence through our bioinformatics pipeline to generate a custom assay. As such, LifeTech sells GMP oligos and enzymes for our diagnostic customers that require analyte-specific reagents. I mentioned in 2010 that we acquired Acrometrics, and so that's the last icon on the lower right. So Acrometrics sells standards and controls. Now the scientists at Acrometrics realized the process of implementing molecular assays in labs can be tedious, with new labs lacking knowledge and existing labs wanting to simplify and streamline their implementation process. My colleague from Acrometrics, Jerry Bunyaratana-Kornkit, will speak tomorrow at 9 a.m. and he will describe the EasyQC Online, a web-based tool for quality control management. And this will help labs overcome these challenges. Jerry will walk through the validation process, providing guidelines and other quality control solutions to assist clinical labs in meeting their verification, validation, and semi-annual calibration verification requirements. To summarize this slide, the FDA clearance of the Quant Studio and the 3500DX represents execution against the company's strategy to become a leader in the diagnostics market by offering both novel clinical assays and best-in-class molecular testing products. LifeTech is aggressively investing in its product portfolio to expand its molecular diagnostics capabilities. Here are two examples. LifeTech has agreements to co-develop companion diagnostics with GlaxoSmithKline and Bristol-Myers Squibb. The BMS Master Services Agreement is for oncology and a broad range of therapeutic areas. The agreement will facilitate co-development that is initiated earlier rather than later in the drug development cycle, from phase 1 and 2 to cover feasibility studies to clinical validation and further development, commercialization of companion diagnostics tests. The GSK agreement was signed in 2011 for developing a qPCR-based molecular diagnostic assay for GSK's MAGE-A3 cancer immunotherapy candidate, designed to identify patients most likely to benefit from the therapy. So the LifeTech platform is being evaluated as a companion diagnostic for GSK's cancer vaccine. A multi-gene panel of TaqMan gene expression assays on a TaqMan array card have been selected to support clinical development of candidate immunotherapy for 2 clinical trials. The platform will measure gene expression of mRNA from FFPE samples to aid in the assessment of patients who are more likely to benefit from adjuvant treatment in resected non-small cell lung cancer and also in patients with node-positive melanoma. LifeTech is developing the assay, supporting regulatory approval and commercialization of the assay. The differences in timelines for drug development and diagnostic development is outlined here. It takes a minimum of twice as long to launch a drug and probably costs 1,000 times more. Development of a drug and a diagnostic to go with it depends on collaboration of the pharma with its diagnostic developer. Thus, it's very important to choose the right partner early on. Why is this? The decision point for incorporating a companion diagnostic may begin in phase 2, and the FDA, as you know, will ultimately approve the CDX and the drug together based on their combined safety and efficacy. If it becomes necessary to bridge the assay, that is, if you need to change anything about the assay design, if you need to change reagents, controls, the instrument, then you will need to show that the previous data is the same as what is in the FDA submission. Bridging studies increase risk, and they also make judgments of safety and efficacy more difficult. If there is variability in the bridging between devices, this will add uncertainty to the endpoints. And so, in essence, this may require more samples to be tested or more power, where more replicates are needed per reaction to make up for the variability. In an ideal world, it would begin this collaboration between pharma and the diagnostic developer. It should begin before phase 1 clinical trials, when discovery phase begins with biomarker identification, followed by target identification and validation for selection of the assay, and by picking components which are suited for diagnostic assay development. But this may not be the case. Life Technologies believes in a flexible approach, not a one-size-fits-all, meaning that the basis for selecting a biomarker or target is derived in many ways and from diverse sources. We propose, we support and recommend what suits the pharma customer, whether it is a clinical trial assay, a lab-developed test, or what we call an LDT PMA developed in a CLIA lab, or a full IVD test. LifeTech has strategic partnerships with CROs, which provide a diversity of support depending on the requirements for the project in question. A lot of times pharma researchers come to us with feasibility data. The data may be from in-house labs or it can be from a CRO. They show that they've identified a biomarker, often with our technology, which is a potential companion diagnostic for the drug. So pharma has this hypothesis that the genetic alteration, whether it is a mutation, a gene amplification, a change in relative expression, that is predictive of a therapeutic response and thus selection of patients for treatment. Our job is to design an accurate and cost-effective method to identify this genetic alteration. These critical decisions early on can determine the likelihood of success with respect to platform selection, assay development and design, and regulatory and quality strategy. The selection of a partner with the end goal in mind reduces risk. As a drug developer, we strive to design a prototype. And so going through feasibility to clinical validation, this prototype needs to align with the drug developer's timeline. Once it's decided that a companion diagnostic test is needed by a pharma company, our goal at LifeTech is to understand as much as possible so that we can propose a workable, flexible solution to our customer. These questions help us make recommendations on next steps. What assay are you using? Was it built from scratch? Are multiple biomarkers being detected? How was the test designed? Is it one of our off-the-shelf assays? For assay design, are there preliminary screening questions employed? In other words, is it a standalone test or is it one of a series of tests? If the assay design requires optimization, or if the assay design is needed for a novel mutation that we do not yet have as a research use only assay, it is relatively easy to generate new TaqMan assays using our bioinformatics pipeline. This actually plays to our strengths in that we have developed at LifeTech many of the technologies which are used on our instruments. Other questions. Does the scientific literature support this biomarker? This has to do with its clinical utility. Is it potentially useful for the patient? What are the clinical findings? Is the clinical trial set up to study the markers? We need to know answers to questions about the disease or type of disorder. What is the specific clinical disease? In oncology, has the test been applied to a specific subtype of cancer or just to solid tumors in general? What are the clinical findings defining this disorder? What is the prevalence of the marker or mutation in the specific type of cancers? Are there epidemiologic factors such as smokers versus non-smokers, ethnic association, age, etc.? All these are important for us as a diagnostic developer when considering how easy or difficult it will be to acquire clinical samples needed for development and clinical testing. Is the test new to the FDA? If a similar test platform has been previously cleared, this reduces risk to the submission. And then, finally, IP issues. Will we need to license the IP in order to make a diagnostic test? More questions. What does your current assay look like? So typically when we are exploring a new assay, we look at 5 components. So they're listed here in detail. So basically it's sample, the assay, the instrument, the software, and the controls. So for sample processing, one has to consider sample collection, sample storage, sample prep, sample extraction. We have a number of commercial kits which are available for nucleic acids. Ambion has RUA products that are an ideal starting point for sample prep. For assay, I've already discussed this in some detail with TaqMan and Sanger sequencing. In the future, we hope to have our next-gen sequencing as an assay platform for companion diagnostics. Third, I've mentioned Acrometrics, who builds controls for both infectious disease and oncology with GMP. I've talked a lot about our instrument platforms, those that have 510(k) clearances for qPCR as well as for CE sequencing. And then we also build software that goes with our instruments for both primary data reporting and interpretive software for analysis of the primary data. Of course, the million-dollar question is, can your assay be successful? So what is needed to make an assay? It's not just the scientific aspect. There are commercial aspects. So there are challenges to profitability, such as low test volume, low reimbursement, or maybe even lack of reimbursement. And then one has to ask whether there are other assays on the market which are equivalent to the assay being proposed. With respect to the markers, what is the ease of use in the lab? And what's the practicality? For example, if sample is limiting, for example, with non-small cell lung cancer, are there workarounds? Can preamplification be used? What's the footprint in the lab? Are there sufficient diagnostic capabilities and infrastructure for launch? Specifically, is the install base for the instrument adequate? Then there are issues around sales. Who can sell it? Is there a global sales force in place? So possible obstacles to success are, what if you have a commercial presence in the countries where the test is to be used, but the commercial force is not deployed yet for the target market? There has to be some training done in order to target the market. And then what about opportunities for co-promotion of the drug with the diagnostic test? Also, together, the pharma with the diagnostic partner needs to plan for regions with thought leaders, those who can enable the test to be deployed. Market development activities need to be planned. Educational materials need to be developed and validated. And then I also mentioned countries, so country-specific distribution channels, as well as a chronological launch plan. There is a need for specific, steady, and regulatory requirements for certain regions in the world. I mentioned training for sales reps for call points. So the goal here is to maximize the benefit of testing for effective use of the drug. Service and supports required for the system, not just for the instrument. And again, as I mentioned, reimbursement. Is there economic data to support reimbursement? Okay, what about issues or bottlenecks or speed bumps? What can you do to streamline the diagnostic development? We know there is risk with drug development. Are we making it any less risky by adding diagnostic development? Hopefully the answer to that question is yes. So the first question here is, what if your previously ubiquitous drug serves only a subset of the target population? Well, this may not be an issue. With the Pfizer drug Xalkori, only 2% of non-small cell lung cancer patients have the ALK translocation. However, the response rate for this drug was 57% when patients had translocation-positive lung cancer. And in fact, this drug received accelerated approval. What if the biomarker set doesn't meet expectations? So again, here we're looking at positive predictive value and negative predictive values, where a positive test reflects the underlying condition being tested for. That is, the marker, such as a mutation, indicates that the patient will respond to a drug. What's the certainty of that? On the other hand, a false positive is the event that the test makes a positive prediction and the patient has a negative result under the gold standard, i.e. that the patient has the mutation but does not respond to the drug. What if your assay contains RUO reagents? This may or may not present a problem depending on the timing and the complexity of the reagent. If pharma plans to turn an RUO assay into an IUO or an IVD assay, then the components of the system must be examined for any issues related to this. And hopefully, bridging studies are not required. If the design history file is not complete, well, this depends on how complete or incomplete, and on a case-by-case basis we can work through an issue such as this. And then finally, do I need a bridging study? Hopefully not. As discussed, we want to avoid bridging when, if at all possible. Okay, this is my second to the last slide. How can you work with a diagnostics partner? It's easy to say, have a plan. Careful consideration of the development and commercialization objectives is very important. Structuring the deal and the milestones to match the risk, not only for the drug development, but also for diagnostic development is important. And then you want to control your choices. So once your prototype has been selected in feasibility, you want to stick as close as possible to that without making additional changes. You want to assess all of your assay components early, and it's very important to have good communication with your partner, as well as with the FDA. And they encourage pre-submission meetings early and often. So in summary, regulatory expertise and commercial infrastructure are very important. Our key conclusions are shown here. Life Technologies partners with pharma to develop companion diagnostic tests. So when selecting a companion diagnostics partner, ask, what are your development and commercial goals? LifeTech has many IVD platform options in sequencing and real-time PCR. However, LifeTech provides more than just technology. We have a number of strategies, as I've described. We encourage avoiding changes that can add unnecessary expense or delays. We have expertise in product development, as that's been our core business for many years. We have experienced product development for IVD, and we have access to development of LDT PMA assays through CROs. And in addition, we can work with our CROs through clinical validation, regulatory, and manufacturing. Our global commercialization capabilities are already in place, and as I mentioned, we have CRO partnerships that support custom needs. We have a regulatory group that provides support to all of our molecular diagnostics projects. And finally, we have a commitment to targeted therapies, personalized medicine, with a strong presence in medical diagnostics and genomic medicine. So thank you all for your attention and attendance today. And I'm available to answer any questions you might have. Thank you very much.